Okay, I think that's enough time. So I guess before we start, just for you to know, the exam will be open tomorrow and you'll have an entire week to complete it. It's about 30 questions, multiple choice, and if you're attending right now, you do get extra credit. I just ask that you participate. Like if I ask you something, just type in the chat or unmute yourself. And then for my students that are not able to attend, I had the alternative option that you can just write a summary and then I'll have a place for you to upload it within week 7, and then that's completely fine. But if you are here at the end of the session, I'll just ask that you type your name so that I can I guess I can write it down and then I can give you some extra credit points. And then I think that's really it. So let's go ahead and get started. Hopefully we can get through this within an hour. And Jabetsi, you can't hear. Does anybody else have an issue with audio? No? Okay. Well, she left. Hopefully when she comes back, she can hear. Okay, so for the first chapter, chapter 4, this is about cells, cell structure, and cell function. So to start off with cells, there's a few people that are really important as far as who discovered cells and I guess the parts of the cell. So for as far as the first person that observed them that we know of, that's Mr. Robert Hooke. He looked at a cork under a microscope, and so he is the one that we give credit to first mentioning or bringing attention to cells. And then we have these two other people that gave us the cell theory, which has 3 parts. That cells come from pre-existing cells. Whoa, Jabetsi. Jabetsi, can you hear us now? Well, hopefully she can kind of hear us. But okay, so let's go ahead and move forward. As far as types of cells, there's 2 basic types that we go over. And let me put them in the chat, like prokaryotic and eukaryotic. So those 2 words, prokaryotic and eukaryotic. And can some of you all tell me, well, let's see, let me put it in the chat. Let's see. What are some differences between prokaryotic and eukaryotic cells? And so you can either answer it in the chat or you can unmute yourself.
Can I answer that question?
Sure.
Sure. Okay. So eukaryotic cells, they have a membrane-bound nucleus, and the prokaryotic cells do not.
Okay, good. Thank you. That's true. And then somebody else said size. That's true also. The eukaryotic cells are much larger than prokaryotic cells. Prokaryotic cells have less organelles. Like you won't find a mitochondria, you won't find a chloroplast, you will not find Golgi Apparatus. So they have less organelles, they're smaller. They still do have genetic material. They still do have a cell wall, they have a plasma membrane. They can have flagella. So they do have some similarities, but there are some differences as well. On this slide, they talk about the nucleoid or the nucleoid region. So for a prokaryotic cell, they don't have a nucleus. They have what we call a nucleoid region, which is just a centralized area where they keep their RNA or their DNA. They do have a cytoplasm. And for the cytoplasm, yes, it's the like jelly that holds organelles together, but there's also a metabolic process that happens there, which is glycolysis. For ribosomes, the word synthesize just means to make. So for ribosomes, they make proteins. And so proteins come in different forms. Most importantly, what you'll probably need to remember is that proteins are large molecules, and a really good example of that could be like an enzyme. So enzymes are proteins. And then the plasma membrane is what you would see on the outer part or the outer region of the cell. And we'll talk more about that. There's a whole chapter on that, so you'll get more info on that. So on the screen, you've got a picture of a prokaryotic cell. It has a flagellum, a singular one, but they can have multiple, like you can have them on opposite sides. And they do have what we call pili, which is similar to cilia, but this is a much smaller form. So they have pili, and they use the flagellum for movement. Pili, they can actually do this. They can use this to extend it out to another prokaryotic cell and do, they can exchange genetic material, which is kind of cool. But yeah, so most common examples for prokaryotic cells would be things like bacterial cells. Annalise, yes. Can you not hear me, Annalise? Let me put it in the chat. And then for this picture, I have a picture of an animal cell. Can you all put in the chat, how do you know that this is an animal cell and not a plant cell? Okay, good. So there's no cell wall, so very good. So this has more of a round shape opposed to like a plant that has more of a angular or square shape. And then for, let me see for Annalise. Annalise, let me just tell her. Okay. And then also, another thing also is that there's not a large central vacuole in the middle of this cell. For plant cells, they will have a cell wall, and then they'll have a really large central vacuole, which I think is the next picture. There you go. So you've got a large central vacuole in the middle, and so that is what's supposed to be storing water as well as food for the cell. But they do have a lot of similar things, like the plant cell and the animal cell both have the nucleus, they both have the rough ER and the smooth ER, they have a Golgi apparatus, mitochondria. Now, the chloroplast is something that is unique to the plant cells. In an animal cell, you will not have a chloroplast. You do have mitochondria, though. And I think that is really it for as far as differences. So, the cell wall, I think we had mentioned it in chapter 3, just what it's made out of. It's made out of cellulose, which is just a really, really hard carbohydrate. So as far as the nucleus, it does hold the genetic material, yes, but it also has these pores on the outside, and so the pores are there because inside of the nucleus is what we call the nucleolus, and that's what makes ribosomes. And so the ribosomes need to exit the nucleus through those nuclear pores. And so then once they leave there, they will go on to the rough ER. And then this one is the Golgi apparatus, which it sometimes is mistakenly looking like the smooth ER. They do look similar. But the Golgi apparatus should be farther away from the nucleus, and the structure, or not the structure, but the function of this is to sort and transport proteins. That's really its main purpose, sort and transport proteins for the Golgi apparatus. Another word for Golgi apparatus is Golgi body. If you do see those, they're the same thing. For the ER, there's two types of ER, the endoplasmic reticulum. There's the smooth and the rough. The rough ER is always going to be closer to the nucleus, and it has ribosomes all over it. And those ribosomes we mentioned earlier, those come out of the nucleolus, then through the nuclear pores. And then outside of the rough ER is the smooth ER. So the smooth ER is in charge of making lipids or fats, and then the rough ER has those ribosomes which make proteins. As far as the lysosomes, these are typically known as, I guess, organelles that clean up the cell. If there's anything toxic or if there's something dead inside of the cell, this will come and clean it up. In the picture, they have a dead mitochondria, which that happens. Like organelles they'll die and then they'll need to make new organelles. So instead of this organelle just being rotting inside of the cell, the lysosome will come and it has digestive enzymes to break down that organelle, and then it sends it out of the cell. But so that's the purpose of the lysosome. It's the one that cleans up the cell with its digestive enzymes. As far as plant cell walls, there's another thing that we should really talk about is because they have this really strong structure, how is it that they're able to exchange water and exchange food with its neighboring cells? And so what they have is they have these channels, what we call the plasmodesmata. I'm not sure if I have another slide on that. I mean, I might, maybe. But the word, it's kind of on the upper part of the picture, plasmodesmata. So those are just channels. They're kind of like little highways within the neighboring cells, and that's how they're able to exchange water, exchange sugars, whatever it is that needs to be passed through. That's how they do it, is by having these little tiny openings that we call the plasmodesmata. And then we already talked about the vacuole, so I think we're good there. This one too is something that I really need you to remember, is this what we call the Endosymbiotic Theory. There's these two organelles that we know used to be their own cell. And so, and we know this to be true because they have these properties, such as that they have their own DNA. So, and the two that we're talking about are the mitochondria and the chloroplast. Chloroplasts and mitochondria both have their own DNA. They have their own outer membrane, and then they also metabolize on their own. And so that's one of the distinguishing features for something to be considered to be living, is that they have to metabolize. And so a chloroplast can metabolize on its own, as well as a mitochondria. And so it's because of these traits that we know that these used to be their own cell at one point, that they were both prokaryotic cells, and then they were, somewhere along the line, they were engulfed by an animal cell or a plant cell, and now they just kind of live there in a symbiotic relationship forever. So, but just kind of keep that in your mind, that you remember the endosymbiotic theory, and that you remember that it's with the mitochondria and the chloroplast. So just make sure that you do review this, because this will be, well, probably everything, well, maybe not everything, most of everything I'm talking about, I promise you, will be on your test. Okay, let's moving on to the membranes. But I guess before that, does anybody have questions? You have questions on cell structure, function, any of those organelles that we mentioned? If you have questions, let me know. We're good? Okay. Let's go ahead and move on then. So chapter 5 is membranes. And I would rather show you a picture first. Okay, let me show you this picture, then we'll go back to that slide. So on the slide, there's a picture of just a singular phospholipid. They have it in its like chemical form, and then how it would look physically. So they look kind of like little jellyfish. And so they have a unique, I guess, a property to them, that they have both a hydrophobic part and a hydrophilic, meaning water-loving, and then a water-hating part. So the head is what we call a hydrophilic head. That head is, it's okay with water, has no issue, but then it has these hydrophobic tails, which are water repelling. And so that's why when you look at a cell membrane, which is this picture, when you look at a cell membrane, the tails are all facing inward because they're hydrophobic and they do not like to be around water. We also call that they're non-polar. So anything that is, let me put it in the chat, anything that is hydrophobic is non-polar. Hydrophobic is non-polar. Hydrophilic is polar. So I have that in the chat for you to remember. So those hydrophilic heads are polar, meaning that they're okay with being around water, and the tails are hydrophobic. So that's why the cell membrane is oriented in that way, that the heads are on the outside and that the tails are on the inside. But yeah, so this is a phospholipid, and let me go back to that first slide where it says that they're arranged in a bilayer, meaning that there's 2 layers of phospholipids, and then you can have proteins that are within the lipid bilayer. And then also we call this the fluid mosaic model just because it is what it says, it's fluid. So there's no, it's kind of, imagine it kind of moving maybe like the ocean, like it's just this waving kind of moving thing. But again, we call it the bilayer just because there's 2 layers of phospholipids and there's proteins embedded within it, and all of these proteins in this picture have different functions, and we'll kind of go over those in a little bit. So, well, let's look at, we'll read these and then we'll look back at that picture. So some of them are for transportation, some are enzymes, some are cell surface receptors, some are identity markers, some adhere or are adhesion proteins, and then some attach to the cytoskeleton. And so if you look back on this picture, you can kind of see which ones are which. So like these down here, peripheral proteins, and these that attach to the cytoskeleton. So like an animal cell, although it's not as rigid as a cell or as a plant cell, it does still hold its round shape, and it's because of these proteins that hold on to the cytoskeleton. Hence the name cytoskeleton, just like you have a skeleton in your body that gives you structure. It's the same thing within a cell. The cytoskeleton does help provide that structure to an animal cell, even though it doesn't have a rigid cell wall. And then these glycoproteins, they'll be used as cell surface markers, so cells are able to communicate with each other, not in the same way that like you and I can communicate, but they're able to send signals to each other, and so this is one of the ways that they can do that. Okay, and then this, I guess, is just another picture of the multiple proteins that are on the cell membrane. And notice how they're large. Proteins are always, I think we had mentioned it earlier, they're large molecules, so proteins are always large. And they take a lot of energy to make. They're not something that's relatively easy to make, so your body will not just be creating proteins unless it has to, right? Your body is efficient in that sense. It will not overproduce what it doesn't need to. So there's 2 types of ways that, I guess, within a cell, how stuff is moved around. One of the ways is without energy or without ATP, and one does require energy, which is in the form of ATP. So for passive transport, let me just put it in the chat. Passive transport is no energy. I'm going to put ATP. Active transport uses energy, ATP. Okay, so for passive transport, it doesn't use any energy, and the way that it's able to move things is by going with the concentration gradient, going from high concentration to low concentration. And the word that they have there is diffusion, which I'm not sure if I have a picture of that. I think I do. Yeah, okay, perfect. So for diffusion, going from somewhere that is a high concentration to low, so like in the, first picture, there's somebody dropping, I guess it's like food coloring or food dye, I'm not sure what it is. But if you notice in the second one, it's super concentrated in B, and then over time it disperses. So that's what we mean by high concentration to low. Similar things with like scents. That's why like in hospitals they're super cold. Well, that's one of the reasons. I'm sure there's multiple reasons. One is like so that it kills bacteria because it's cold. Another one also is scents. Scents are really amplified when it's hot versus when it's cold. And I'm sure you've noticed that too. If you're ever somewhere where it's hot and somebody smells bad, I promise you it's going to smell so much worse if it's hot versus if they smell bad and it's cold. So that's kind of how it works. And that we call that diffusion. But that doesn't require any energy. Another one also is osmosis. Osmosis is also a form of passive transport. So this again doesn't use any energy, no ATP, and osmosis is the movement of water. So water going from a high concentration to low. Water always wants to be equal. There's some words that maybe you've heard like homeostasis or something like that. That's essentially the same thing. So water always wants to be equal. And so it's able to do that from going from high to low, and we call that osmosis. For active transport, we already mentioned it. This does use energy, which is in the form of ATP. And usually active transport will require the help of proteins. And I think I have some pictures for y'all to look at. There we go. Right, so this process, I have two on the screen, but you can kind of tell like this looks like it would need more energy, or it would need energy, versus just moving water through a cell membrane. That's not going to use any energy. But if you're moving large particles inside of the cell or outside of the cell, then yes, you're going to have to use ATP. And the two examples that are on the screen are called endocytosis or exocytosis. It's a type of bulk transport, just meaning that it's large molecules. So endo means in, so going inside of the cell, and exo means out, going outside of the cell. There's other words too, pinocytosis and phagocytosis. So I'm going to put those in the chat. Pinocytosis is what we call like cell drinking, which is just fluids. So if there's lots of fluids coming in, we call that pinocytosis, and the other one is phagocytosis. Cell eating. So phagocytosis is intaking large molecules, which is the endocytosis picture, basically. So, but those words are also used, so just kind of take those noted. Pinocytosis, cell drinking, and phagocytosis, cell eating. Yes, these do require energy. So endocytosis and exocytosis do require ATP. Okay, and then for solutions, there's these three solutions that I guess determine the, well, for plants we call it turgid, if it's turgid or not. And then for regular cells, it just, I guess, influences the cell membrane. So the three are hypertonic, isotonic, and hypotonic. Same for the plant cells. They're just the only difference between the animal and the plant cells is that you notice the cell wall in the plant cells, it doesn't allow the cell to move as much as the animal cell. But if you notice on the plant cell that's in the hypertonic solution, which means it's super salty or saline, it does shrivel up and pull away from the cell wall, and we call that plasmolysis. But yeah, so hypertonic means that it's going to be in a like salty solution and it shrivels up, or that it's just doesn't have any water. So it could be one of those two. So hypertonic is maybe it's in a salty solution, or that it's lacking water and it starts to shrivel up. Isotonic is when the cell is happy It could be maybe that there's enough water outside and inside of the cell, and so it's just a very happy cell. And then for hypotonic, it's swollen, and then for a plant cell, it probably will not burst. But for an animal cell, yes, it could burst, and we call that lys. That little lysis, I believe. Let me see. There we go. Let me put that in the chat. Why what? Why, Victoria, go ahead. That's okay. You can type it. Let me go to the next slide. But go ahead, type your question. I'm not sure if Victoria, if you had a question on the hypotonic solution. So that can happen if there's not enough salt in the water. So like regular water does have salt in it. It has minerals. And so that, I guess those are certain properties that maybe that cell is used to. But if you put it in something like distilled water, which is like pure water, too much water will go inside of the cell, and then it'll cause the cell to get really large, and then it could eventually pop. Yeah, it does kill it. So we're on to our last chapter. Are there any questions so far on cell membranes, anything like that? Active transport, passive transport? And I'll also post the slides. If you all want to look at the slides later, I'll post the slides as well. So the last one is Chapter 6, Energy and Metabolism. We mentioned earlier that for something to be considered to be living, it does need to metabolize. So the two forms of energy that I guess we refer to are kinetic and potential. And so we kind of define energy as the capacity to do work, and the two types are kinetic and potential. So the girl at the top of the slide has potential energy, and it's not until she actually goes down the slide that we call it kinetic energy. So potential is stored energy, and kinetic is the energy of motion. And so there's different forms of energy. They give you some examples in the next slide, like for a calorie, right? That's like for us, how we get energy. So like for us, we're not able to create our own food. We're heterotrophs. We have to eat. And so the way that we are able to get energy is by eating. And sometimes we eat too much, and then we just have all this energy inside of us that's stored. So it's kind of this battle that we have. But so calories can be a form of energy. And so here it says that a calorie is heat required to raise 1 gram of water 1 degrees Celsius. And then one of the main things, and I have it bolded in this slide, is like, where does all the energy in the earth come from? And it all does come from the sun. And then all that energy is just kind of diverted elsewhere, right? So the sun provides energy. Plants are able to capture it. I do like that it says photosynthetic organisms, because there's other things that can photosynthesize. Like there is photosynthetic bacteria. So that's kind of, I'm glad that they mentioned it in that way. So anything that can photosynthesize captures that energy, and then that energy can get passed on. Either like you eat the plant, or an animal eats the plant, and then you eat the animal, or however that wants to work. What is a vacuole? A vacuole is the organelle inside of, it could be in an animal or a plant cell, and they store water and food. And I think that's it. Any other questions? You're good? Okay. I think that we went over this in chapter 2, but just kind of as a recap for these words, because they are tricky: oxidation and reduction. When something is reduced, that means that it's gaining an electron, so it's kind of the opposite. So reduction is to gain an electron, and oxidation is when you're losing an electron. And so when you have what we call the redox reaction, that just means that they're happening simultaneously, that you have this exchange of energy simultaneously. But I wanted to just go over that again just because that one is tricky. And that word reduction, make sure that you know it because it will be on your test. So reduction means that you are gaining an electron, gaining. Yes, it's the opposite. So there's 2 laws for thermodynamics that we have to go over. The first one is probably the easiest one. I think most people are familiar with it. It's that energy cannot be created or destroyed. It's just like we mostly talk about the flow of energy. So the energy is coming from the sun and then it's just kind of transferred everywhere. Like maybe you remember in elementary they made you learn the food webs, like that it goes to the plants, the producers, and it goes into the primary and the secondary, and then there's decomposers. So the energy is not being lost, it's just being moved around. And so that's how that works. For the second law, entropy is continuously increasing. Energy transforms proceeds spontaneously to convert matter from a more ordered, less stable form to a less ordered or more stable form. And then for this slide, there's kind of 2 routes that they're showing you. The word reactant, if you're familiar maybe in your chemistry class, the word reactant and products. So like at the beginning of an equation, those are called the reactants. And then it yields a product. So, but the reactants is what you start off with and the product is the end result. So for here, the uncatalyzed is taking much longer to get to the final product versus something that has a catalyst. And so does anyone remember what a catalyst is? What is a catalyst? Anybody remember that? Very good. Very good. So a catalyst does speed up the reaction, and also a catalyst is an enzyme. So a catalyst is an enzyme, and enzymes are proteins. Right, and so we'll talk a little bit more about enzymes in a second. But so for this reaction, if it does have a catalyst involved, it's going to get to the end result much faster. That's kind of what this is showing you. Also, because we're talking about energy and metabolism, we did talk about ATP before, but I'm not so sure that we kind of know where the energy comes from within ATP. So ATP is adenosine triphosphate, meaning there's 3 phosphate groups. And so the energy comes from when the last phosphate group is released. And so ATP turns into ADP, and that energy is given off, and that's what helps power the cell. So you'll have this, and they call it a cycle, where ATP turns into ADP, but then phosphorylation goes, and it's just this continuous cycle. ATP, lose a phosphate, becomes ADP, gains a phosphate, and so that's just how it works. But ATP itself is not energy, it's just when ATP loses one of its phosphates group that it does release energy. And then here we've got our enzymes. How does the water come in? ATP plus water, energy for endocytic cell. So, like for photosynthesis, there's some parts where the H2O will give off a hydrogen and it'll reduce, And so that it kind of plays into electrons. It kind of gives off electrons, and so that's how it's able to regain its last phosphate group. Similar to when we will talk about, we haven't gone over it yet, cell respiration and photosynthesis, you'll see that where the water will split, we call it the splitting of the water, and those hydrogen ions supply electrons. And so that's how that kind of comes into play. For this picture, the enzymes, not sure how familiar you guys are with like food breaking down, but you have enzymes inside of your stomach, and then there's some that you don't have, which is kind of interesting. So like, does anyone know what's it called when you cannot digest milk? What is that called? When people cannot digest milk? Yeah, very good. So lactose intolerant. So what that means is that they do not have the lactase enzyme, which is required to break down the sugar. So let me put it in the chat, lactase. Oh, and another thing, if you see a word that ends with ase, more than likely it's an enzyme. Lactase is an enzyme. So anything that ends with ase, which I don't see any other ones on this screen. There's another one, sucrase. Sucrase, right? So anything that ends with ase is an enzyme. So what the enzymes will do is they break down larger molecules. So like you're saying a polysaccharide, yes. So in this picture, it's not a polysaccharide, but this is a disaccharide. And we know that because we see two. So this has two sugars put together, so it's a disaccharide. And so your body cannot digest disaccharides or polysaccharides. It can only digest sugars in a singular form. So if you look at the end result, it breaks it down, and then you have two singular sugars at the end. And so for people that are lactose intolerant, they don't have the enzyme to do this. They don't have the enzyme to be able to break down the sugar from milk. And so therefore they have stomach aches, I think. I actually don't know. I think they get stomach aches. I don't know if they throw up or anything. I'm not so sure. But I just know that the body rejects it, and so it's because they're lacking the enzyme. I'm not sure if anybody... Oh, diarrhea and bloating. Oh, that sounds awful. I don't know. I don't eat dairy, so I don't know if I'm lactose intolerant though. Cramps. I know you can. I think my friend buys like the enzyme. Like you can buy it at HEB. I can just eat the tablet, and you can buy the pill. I don't know how much it is. I'm pretty sure it's relatively inexpensive. But so there's ways to get around it. If you are lactose intolerant, you can just take a pill, which I guess is just enzymes, so that it can help you break it down. So yeah, some people are able to drink dairy and it's fine. If you kind of look that up, it's inherited, I think, from European descent. And so some people are able to drink milk. Most people, I don't think, can. And so if you can't, that means that you didn't inherit that it's a mutation. But I'm not sure. It happens with age. Possibly. I drink milk like water. Oh, okay. Yes, it's common in Hispanic and Black descent. So yeah, like we said, if you're able to drink milk, that's a European descent. And maybe you're right. Maybe with age, it does happen where it's just worse. To be honest, I'm not so sure. I don't ingest dairy. But okay. So things that can affect how an enzyme works. There's quite a few things. Probably one of the main ones is temperature. Temperature can either help bring an enzyme to its optimal, I guess, workability, and then as well as pH. So the pH scale is from 0 to 14, and so enzymes, and it depends on the enzyme. And I think I have a picture. Let me see. Yeah, okay. So on the screen we have different enzymes, And you can see that they peak at different, and for the top one it says temperature. So like this enzyme, optimum temperature for a human enzyme, it doesn't say what it is, but so for this specific human enzyme, it works best at 40 degrees Celsius. And then anywhere else, it's not going to work as well. Just the same thing over here, it says optimum temperature for an enzyme in the hot springs. It works best at 7 degrees Celsius. And then anything outside of that temperature, it's not going to work as effectively. So that's the same for pH on the bottom. They show you too. Like for this pH for pepsin, it doesn't work so well outside of the range of 2 to 3 pH. And so that's really all that they need you to know, is that they're very specific, or they work optimally in a good pH range or at a temperature. But that's not to say that all enzymes work at a specific temperature or pH. And then another thing too is that they can, let me go to this picture. So like this blue thing is the enzyme, which is a protein. They can change their shape if it gets too hot or if it gets too cold. The actual enzyme will change shape, and because of the shape change, it's not able to get these reactants and break them down. And actually there's a word for that. Let me put it in the chat. Denatured. Right, so enzymes can become denatured, meaning that they're not going to work as properly, and that can be from extreme temperature changes or extreme pH changes. So like for your hair has certain proteins, and that's how it looks the way it looks. Like some people have straight hair, some people have curly hair. And so like me, if I had really curly hair, I could change it by using a flat iron. I could straighten my hair, and then it'll be straight, but it won't stay straight, right? Once I wash my hair, it goes back to curly. And so that's a really good example of how proteins can go back to their normal form. So like because I applied heat to my hair, the proteins do denature, but then once I wash my hair and they go back, they're able to go back to being, I guess, the curly form. So just know that. So just know that enzymes, they can change their shape, and we call that being that they're denatured, but they can always go back. So that's kind of a cool thing. And then this is probably one of the last slides. Oh, with no hair. Well, then grow your hair. Grow your hair and try it. So for one of the last things as far as enzymes, we have these two types of inhibitors. One is called the competitive and the other is the allosteric. So the only difference between these is the location. For competitive inhibitors, they're going to block what we call the active site, which is on the, I guess, the top portion of the enzyme. And the allosteric site is on the bottom part. And so what the allosteric site, I guess once you have an inhibitor go into the allosteric site, the active site will change shape, and then the substrate is not able to go in. In the other one, the competitive, you can see that the substrate is not able to latch onto the active site because something is in the way, versus in the allosteric site, there's nothing in the way, but the active site changed shape, and now the substrate cannot lock on. And when they lock on, we call that an induced fit. So, and I think that is it as far as my review. An example of an inhibitor. So in the slide, it's going to be this green thing as far as like what it actually is. I would say, what are they called? Promoter and repressor proteins. So your body can create what we call repressor proteins that, like similar to turning on and turning off genes, like we mentioned that your body is like very efficient. It's not going to do something if it doesn't need to do. So like for here, this enzyme could be maybe breaking down, I don't know. So we can go back to the milk example. So like if you were to drink milk, it sends off a signal to your body to break it down. And so for this enzyme, if it's not going to be used, like if there's no milk in your system, there's no need for that enzyme to be working or to stay open, so that an inhibitor will come and kind of block it so that it's not working when it doesn't have to. So that's kind of how that works. So it's very interesting how your body does that. So any other questions that you all have? It could be outside of what we reviewed today. That's a good question. I will probably do it this weekend, but thank you for reminding me, because we're at the end. Could you all type in your names? The test is due not this Friday, but the next Friday. So I guess before you leave to spring break. Yeah. Type in your name, please. Type in your name. And then Amanda, let me see. Once you type in your name, you can go ahead and take off. You're good. The test will be open tomorrow, and then you will have a whole week for that. So as soon as you type in your name, you can go ahead and take off. The recording will be available. So, I mean, I don't know how long it takes to upload. It'll be available for sure tomorrow, or maybe, I guess, in the middle of the night. I'll just leave it to be uploading throughout the night.